So our first speaker in this second session is Professor Vivian Mushahwar. She's a professor in the Department of Medicine, specifically Division of Physical Medicine and Rehabilitation at the University of Alberta. I'm letting that audio fade away, hopefully. She directs the SMART network, the sensory motor adaptive rehabilitation technology, SMART. They use diverse approaches to restoring mobility after spinal injury, especially concentrating on using implants to stimulate deep neural networks in the spinal cord. Dr. Mushahwar.
Thank you very much for the kind introduction, Jay, and special thanks to the organizers for this meeting. It's really a great pleasure to be here today. Now, as Jay mentioned, and as many of you probably know, the overall goal of the work in my lab is to develop interventions that would improve function after neural injury or disease and prevent the development of secondary health conditions. These interventions are anchored in the principles of neuroscience, neural engineering, and rehabilitation. And the focus today, as Jay said, will be on restoring and improving walking capacity in persons experiencing spinal cord injury. But before I start, I would like to acknowledge the incredible group of people I've had the great privilege of working with over the past decade as part of the SMART network at the University of Alberta. And SMART stands for Sensory Motor Adaptive Rehabilitation Technology, and our mission is to develop intelligent medical devices and innovative rehabilitation interventions that significantly improve functional outcomes for people with neurological conditions. And really our goal is to take these innovations to the clinic to improve quality of care and improve patient outcomes while also reducing healthcare costs. Members of the SMART network come from many different faculties, disciplines, and sectors, ranging from fundamental neuroscience to engineering, computer science, rehabilitation, surgery, health economics, and consumer needs. And collectively, the SMART network forms an exciting ecosystem for efficient translation of knowledge into practice and social impact. Now, in my lab, as I mentioned earlier, we focus on restoring function and preventing secondary complications. The primary effort has been on restoring mobility, especially the ability to walk after spinal cord injury. But we also develop wearable technologies to prevent secondary conditions, such as pressure injuries or pressure ulcers, using an intervention we developed a few years ago called Smarty Pants, as well as preventing deep vein thrombosis through a recent intervention called the SOC. Now, turning our attention to walking for the rest of the talk, our goal for decades has been to harness the networks in the spinal cord for restoring functional mobility after spinal cord injury. For conditions with severe spinal cord injury, we focus on reactivating the networks below the injury within this region of the cord, the lumbar enlargement, which contains a large proportion of the neural networks involved in locomotion. Now, to harness the locomotor networks in the lumbar spinal cord effectively, we map the lumbar enlargement by delivering minute electrical currents at different locations throughout the enlargement. And we characterize the elicited movements produced by each stimulated location and developed a map describing the functional organization of the locomotor networks in the cord. We then identify the best locations for tapping into various elements of the locomotor networks. And to summarize many years of work, we found that these regions within the ventral horn were the best locations for eliciting the single joint movements and coordinated multi-joint synergies needed for restoring functional overground walking. So we mapped these particular ventral regions in multiple animal species. And the very impressive findings are that, first, these functional maps are repeatable and predictable between animals within species. What I'm showing here is a 3D rendition of the cat lumbar enlargement, and the colorful regions are the motor neuron pools or the nerve cells that make connections to various muscles in the legs. Now, depending on where we are in the enlargement, we can reliably elicit a single joint movement or a coordinated multi-joint synergy. And this is repeatable from animal to animal. Now, the second impressive finding is that these functional maps are not only repeatable and predictable between animals within a species, but they are also similar between species. And what you see here is the functional map of the lumbar enlargement in a monkey. With only minor differences, the functional map in the monkey is very much the same as that in the cat. This shows that stereotypical networks for locomotion are present in the ventral horn of the lumbar enlargement, and these are preserved between species, suggesting that they are present in humans as well. So these networks reside deep within the spinal cord, which are marked by these blue regions. Now the question becomes, how do we activate them? Now, to do this, we realized that we need to develop a micro implant that allows us to deliver minute levels of electrical current to these regions deep in the cord. This intraspinal microstimulation, as it's called, is similar to deep brain stimulation that has been effectively used to treat various neurological conditions, such as Parkinson's disease. Now, we have developed the microimplant to place in the lumbar enlargement, and the implant consists of ultra-fine electrodes that are smaller than the human hair and a lead that connects them to an implanted stimulator. And this is a close-up of the implant. And what you're looking here is at the surface of the spinal cord, and these are the fine leads that travel on the surface of the cord, with the microelectrodes entering the cord at the end of each lead. And what this implant does is it activates various elements of the locomotor networks in a coordinated fashion, resulting in the generation of functional overground walking. To date, this work has been very successful in preclinical models and has led to impressively long distances of functional overground walking. The figure that I'm showing over here shows the force generated during walking, which is an indicator of the quality and effectiveness of walking, over a distance of 1,000 meters, a kilometer. And what you can see is that the force generated during walking is stable throughout the distance, demonstrating that functional walking was sustained throughout the distance of walking. And this is really an unprecedented distance of walking in a preclinical model of spinal cord injury, especially when compared to other approaches, including stimulation of the muscles or nerves, or even stimulating the surface of the spinal cord using epidural stimulation, the approach that was described by Dr. Phillips in the first talk today. With muscle and nerve stimulation, as well as epidural stimulation, walking decays very quickly after about 100 to 150 meters. So to summarize our findings on the intraspinal micro implant, we are convinced that intraspinal microstimulation taps into organized elements of the locomotor networks and can reliably produce complex functional movements, such as long durations of stable standing and long distances of functional walking. And we are now preparing the implant for first in-human testing involving persons with severe spinal cord injury. Now, in persons with less severe spinal cord injury or persons experiencing other neurological conditions such as stroke or multiple sclerosis, we proposed engaging the arms along with the legs in an activity-based therapy for improving walking to allow for the engagement of the spinal networks throughout the cord. And we strongly believe that this engagement is what is needed for effective walking after various neurological conditions. Now, there's plenty of evidence supporting the engagement of the arms during walking. We already know that the arms swing in coordination with the legs during walking, and this swinging is not the result of the arms just passively moving as passive mechanical appendages. Instead, arm swing is neurally driven, and the neural drive to the arms is coordinated with the drive with the legs. Now, this knowledge has been known for many years, and a number of investigators suggested that the arms can modulate the motor output of the legs. However, the arms have not to date been systematically involved in rehabilitation therapies for improving walking. So we wanted to investigate directly whether engaging the arms simultaneously with the legs does indeed lead to functional improvements in walking, even after chronic spinal cord injury. And to deliver the intervention, we decided to use simultaneous arm and leg cycling as an activity-based therapy that is efficient in terms of staff utilization. This intervention allows coordinated rhythmic activation of the arms and legs. We applied electrical stimulation to the legs to assist with the duration of movement, as well as provide a phasic sensory input to the spinal cord during the movements. We recruited participants with incomplete spinal cord injury classified as AIS C or D. In other words, these participants had some connections preserved between the brain and spinal cord below the injury, but still had substantial disruptions in their walking ability. One group of participants cycled with their legs only, and one group cycled with their arms and legs simultaneously. And training for both groups was 12 weeks long. The primary outcome we looked at was walking speed, which is what you see over here. For this, we used the validated 10-meter walking test. And as many of you would know, in this test, the person is asked to walk in a straight line for 10 meters. They're instructed to walk as fast as they are comfortable to, and the time it takes them to cover the distance is measured. What you can see over here on the x-axis is time in weeks since the beginning of training, and on the y-axis is the percent change in speed over the weeks of training relative to each person's walking speed prior to the initiation of training. The dashed line shows the results from the group that cycled with their legs only, and the solid line is for the group that simultaneously cycled with their arms and legs. Both groups of participants had similar pre-training walking speeds. And as you can see, both groups improved their walking speed over the course of training. And at 12 weeks, at the end of training, both groups showed significant improvements in their walking speed relative to their baseline. But the increase in walking speed was significantly larger in the arm and leg cycling group than in the legs only cycling group. We also saw significant increases in walking distance using the 6-minute walking test. And both groups significantly increased their walking distance at the end of training relative to their pre-training levels. Now, those videos show you what the improvements in walking looked like after the completion of training for a study participant in the arm and leg cycling group. On the left, you see what walking looked like pre-training, and on the right, post-training. Pre-training, you get to see that the participant is hunched over, is looking down, needing a lot of effort to swing his legs and walking fairly slowly. However, post-training, he's standing upright, he's walking much faster than before, and he's no longer needs to look at his feet because he now has restored sensation that allows him to know where his legs are in space without needing to look down. Now, we compared the results of this study to published work involving gait-specific locomotor training, such as body weight supported treadmill training or overground walking training, which focused on the legs only in study participants with incomplete spinal cord injury. The bars that you see here show the increases in walking speed after training for the various studies that had training durations similar to ours, and the dashed line shows the average across the studies. The numbers in the bars represent the sample size for each study. And what's intriguing to see is that leg cycling training in our study was able to improve walking speed to a similar extent to that seen with gait-specific training paradigms. But arm and leg cycling can improve walking speed at least 2 times more. We see similar comparisons here for walking distance. So there are 2 takeaway points to note here. Cycling training translates to improvements in walking, and importantly, simultaneous arm and leg cycling results in twice the improvements in walking ability. And we are now looking into ways to translate these findings to rehabilitation centers where arm and leg cycling could benefit a wide range of persons with neurological conditions that affect their mobility. So to wrap up my talk, I hope that I convinced you that engaging the spinal networks is an effective means for improving mobility after spinal cord injury. Microimplants in the spinal cord can activate otherwise inaccessible spinal networks and restore walking after severe spinal cord injury. Engaging the arms simultaneously with the legs for the rehabilitation of walking significantly improves outcomes for persons with neurological conditions. I'd like to close by thanking the funding agencies and donors for their generous support, without which we would not have been able to perform this work. I would also like to thank the outstanding team of trainees, support staff, and collaborators that have made our work possible. Thank you.
Thank you, Dr. Mushahwar. Really fabulous work. And I have a question about both approaches. I think you said you're about to conduct a clinical trial of microimplants. And I'm just wondering, can you give me, what is that going to entail? What are your experimental or your clinical subjects going to be doing?
Yeah, so just to be accurate, we are now developing the spinal implant that would go in humans. So we're in the process of testing it in an animal model that represents humans. And as soon as this testing is done, we'll be able to apply for regulatory approvals to move to the human testing, the first in human testing. But what that would entail is just that, right? It's similar to what Dr. Phillips described. We need to go through the regulatory approvals. We need to go through Health Canada or the FDA. We need to get what's called an investigational testing authorization in Canada, or an IDE, an investigational design device exemption in the US, to allow us to do this first in human testing. Once we get those approvals, the study would be open for recruitment, and for some preliminary testing that we wanted to do with this, we opened the study for, I don't know, probably a week, and we had more than 50 people expressing interest. For that study, unfortunately, none of them qualified for the study, but we're finding out that we probably will have quite a bit of interest from persons with severe spinal cord injury who could benefit from this. So once the recruitment is done, we will undergo the surgery, and we will then do testing for at least 6 months post-implant to be able to quantify the outcomes and the characteristics of this implant, and the characteristics of the walking that can be restored.
And a question about the simultaneous cycling of arms and legs. And my understanding of the spinal cord might be pretty primitive, but do you understand exactly what mechanically is going on, that movement of the arms somehow helps mobility?
There's still a lot to be known, but what we know so far is quite extensive. We know that there are connections between the cervical portion of the spinal cord that controls the arms and the lumbar portion of the spinal cord that controls the legs. And these connections are called propiospinal connections, right? These are connections in the spinal cord that coordinate the activity of the arms and legs, especially when you're doing rhythmic movements. And we've been perturbing these networks in persons with neurologically intact systems and persons with incomplete spinal cord injury by applying stimulation to the surface of the spinal cord through, to the surface of the skin that sends activation towards the spinal cord. And we could see how you can now, when you modulate the activity of the cervical region, you actually get changes in the lumbar region of the spinal cord, providing yet further evidence that there is nice connectivity between the arms and the legs. And during rhythmic walking, this connectivity of the arms enhances the walking pattern through the legs. And interestingly, it's vice versa, right? The legs enhance the activity of the arms as well. It's just that we're not quadrupeds anymore, so we don't notice it as much. But if you get people to do this to their arms while they're walking, you will still see activity in their muscles, bursting activity, indicating that the spinal cord is sending activity to these muscles, even though you're not moving your arms. showing that the legs also have an input on the arms as well.
Well, thank you very much, Dr. Mushahwar. We will welcome you back for the roundtable in a few minutes. Thanks again.
Thank you.